When we think about biomarker testing, there are lots of different ways that your doctors can do this testing. And this right now, what we're showing you is testing that can be done on the biopsy tissue. So first we have something called FISH, which stands for fluorescence in situ hybridization. What that really means is we know that some of these biomarkers are created when two genes come together abnormally, and they make this abnormal protein as a result. So if you take a probe against one gene and a probe against the other, and they both light up with different colors, then using a special technique, you can see where these two colors merge. And that's when you know that you have an abnormal gene that's present. And this is really important when we talk about certain abnormal biomarkers that are called fusions. Next, we have something called IHC, which is immunohistochemistry. And what this means is we know that there's certain abnormally expressed proteins that can be found on the surface of a cancer cell, and that some of our drugs are actually targeting this specific protein. So what IHC is, it has an antibody that can detect this protein, and then a secondary antibody against the first antibody that can light up. So again, using science and biochemistry, if you put these different antibodies into an assay and it lights up, that means that the protein you're looking for is there. The next option we have something called RT-PCR, which stands for real-time polymerase chain reaction. And here, again, we're looking for a specific DNA change. Now, when I talk about DNA change in this talk, I'm not talking about DNA that is inherited from your parents or that you can pass on to your children. I'm really talking about the DNA that is found specifically in your cancer cells. And here we know that certain biomarkers occur because there's a mutation in the DNA of the cancer cells. And so what RT-PCR can do is we can look for a specific mutation by combining all these different materials into the test tube. But really now, and again, 2025 and beyond, you'll hear your oncologist talk about something called next generation sequencing, or NGS, or molecular testing, or comprehensive genetic testing. What that really means is now we're able to take the cancer cells that we get from the biopsy and put them through a very broad panel, looking through hundreds of different genes that could be present in the cancer cell. And this allows us to detect multiple abnormalities all at once. But beyond tissue, we now have something called liquid biopsies, which are now predominantly taking blood samples. So we, as oncologists, we still think that the tumor biopsy or biopsy of the cancer spot itself is still our gold standard. However, we know that this can be challenging depending on where the cancer is located, how safe it is to put a needle in it, and how much tissue we can get at that biopsy. What we know now too is that cancer cells do enter into the bloodstream, and sometimes their DNA spills into the bloodstream as well. So what liquid biopsies do is you simply go to the lab, you get some blood drawn, and your oncologist sends it to a specialized laboratory where they actually look for presence of cancer DNA in the blood. And as you can imagine, this is way less invasive than getting a tumor biopsy. However, this technique is still fairly young, and so we're not able to detect with 100% sensitivity all the mutations that might be detected on a tissue, but certainly I think liquid biopsies are complementary. And I think there's quite a bit of excitement into perhaps even taking this into the future in terms of cancer screening, helping understand prognosis, monitor response to treatment. I think we'll see a lot more about liquid biopsies in the future. As I mentioned before, tissue biomarker testing and liquid biomarker testing to me are complementary. I think they're both necessary at certain times when we're taking care of a patient. So again, for the tissue, that I think is our gold standard. We're putting the needle right into the cancer itself. And there are certain biomarkers that are really important. For example, the PD-L1 level, which we'll talk about, which we can only do on tissue. However, when we don't have enough tissue, when we're trying to understand how cancer is changing throughout therapy, and we can't continue to re-biopsy, that is where the liquid biopsy really comes into play. And also the results that you get from the liquid biopsy comes back to your doctor quicker in general than the tissue biopsy.